Peltic survey. I suppose a bit of housekeeping, if you wouldn't mind muting your microphones, everyone, just until after the presentation, then we can deal with the questions, and then I don't get distracted, basically. But yeah, so my name is Jeroen van der Kooi. I'm a fishery scientist. I work at CFAS, the Center for Environment, Fisheries and Aquaculture Science and I'm based in Lowestoft. And my work areas are fisheries acoustics, small pelagic fish, their ecosystem, their monitoring, etc. So yeah, quite a broad area. And I'm going to talk about one particular element of that, which is the Peltic survey. So let's get this going. There we go. So this presentation will roughly be about half an hour, maybe just over. I'll start by introducing the Peltic survey itself. I'm conscious not everyone may have heard of it. I'll give you a bit of background about the motivation of the survey, the objectives, and how we try to tackle that, the approach. Then I'll go briefly into a little bit of detail into the results of the last survey, which was the October 2024 survey, focusing in the first instance primarily on the sort of environmental conditions, some of the plankton results, and some of the top predator data. And then I'll go into a little bit more detail into the small pelagic fish community. But I want to focus a little bit. I'll look at 2024, but I'll put it in context of the 12 years that we've been collecting data on those species in the years. And then I'll conclude with a little roundup at the end, including an idea of where you can get more information on some of the papers that I refer to and perhaps some of the data that you may be interested in. Okay, so Peltic stands for the Pelagic Ecosystem Survey of the Celtic Sea and Western Channel. It came about in 2012, roughly, when we had an acknowledgement that there was quite a lot of diversity in small pelagic fish in the southwest, so the waters around Cornwall and Devon. And we believe that one of the reasons why there is such a diversity is because it's an area where some of the cold water species find their southern limit of their range, like herring and sprat. Some of the warm water species, like sardine and anchovy, find their northern range of distribution. So the fact that both co-occur there means there's an extra diversity, which we may not encounter in some other places. Now, most of these, or at least some of these species, were targeted by commercial fisheries, most of which operating from the UK, generally quite small-scale vessels, small vessels rather, and small-scale fleets. So generally perceived to be and thought to be very much sustainable fisheries. But the fact of the matter is, we didn't know because there was very little data available at the time, certainly about the stock status, the population size, etc. So that was one of the reasons why we were interested in that. Now, the other one was that sustainable management of these stocks was particularly important because these small pelagic fish are what we call mid-trophic species. So they sit in the middle of the food web and they channel energy from the plankton all the way up to the top predators. And some of those top predators include, of course, the important marine mammals, seabirds, but also commercially important whitefish, like things like hake, etc. So really important that we have a good idea of how many fish there are and that we manage it sustainably. The other thing to note about small pelagic fish is that they're very short-lived and they're highly productive, right? So they don't live very, use loads of eggs. So certain conditions that are favorable to the survival of the eggs will mean that from year to year, there can be a huge boom and bust in the population size. And that's particularly interesting because of the area where it is. So I already mentioned it's at the boundary between the colder and warmer waters, and therefore species that are favored by cooler temperatures will do better during cooler periods, etc. So the area, we believe, and the species they're in are of interest because of the potential use of these species as climatic indicators, basically. So we knew that just simply counting the fish for any stock assessment wasn't going to be enough. We really wanted to understand what drove some of the patterns we were expected to see. And for that, we needed to study not just the fish themselves, but also their wider ecosystem. So the other thing I wanted to point out is that we chose to sample the area in the autumn. A number of reasons for it. One is the timing of the fishery. So both spread and sardine fisheries start roughly then. I mean, maybe a little bit earlier, but you can imagine that sampling or measuring the size of a stock after the fishing season is not as helpful because there's likely to be removal of fish. So you do want to do it earlier in the fishing season instead. The other reason that we focus on the autumn rather is because that's when we learned that spawning sardine reside in the area. So it's an important spawning period. And according to the Coombs et al. Paper from 2010, it's becoming more and more important compared to the earlier spring spawning peak, which was recorded previously. Now, one final reason which is associated with that spawning period is that because there is known to be an autumn bloom, which is of relative importance, we wanted to understand how the productivity created by that autumn bloom would help survive the larvae of these autumn spawners through the winter. Right. So this is a map of the survey area. You recognize the Cornish Peninsula. And what you see here are these black lines, which are what we call transects. And then these red and yellow stations are our hydrographic stations. So effectively what we do every autumn, we steam with our research vessel along this transect during the day, and we collect acoustic data. So this is echo sounder data, which maps and quantifies the small pelagic fish community. Now we supplement that with small trawls, where we take samples of the fish to understand what's there, but also to collect biological data of those fish that we're seeing. Whilst we steam, we also have a couple of other continuously sampling instruments. The top left picture is a picture of what we call the ferry box. So it pumps water just below the sea surface and uses a lot of instruments to measure temperature, salinity, chlorophyll, primary production, things like that. The other thing we've recently started to develop is with colleagues, both here and at PML, is what we call a plankton imager. So another flow of water from the subsurface gets pumped on board, and at a credible rate, pictures are taken of all the plankton that are found there. Okay, so at those red and yellow stations, we then stop at night, and then we deploy a couple of other instruments. So the top left is our Rosette CTD system. It provides us with a vertical profile of the water column. So all sort of oceanographic information like thermocline and other features are recorded with that. We can also collect water samples, which we use to calibrate some of the sensors. Bottom left are different mesh size plankton nets, and we sample the plankton community with that, not just the potential prey like the copperpodes, but also the ichthyoplankton, the larvae and eggs of particularly sardine that I mentioned earlier. Now, interestingly, both the Rosette and the Niskin bottles, where we collect water samples, and the plankton samples have been providing us with extra information. So we've been working with environmental DNA recently and exploring how we could potentially use that in future monitoring. That's what this helix is about. And then the two pictures here are perhaps less good news, which is unfortunate litter that we encounter. So we measure macro litter in the trawl, which is that center picture, but increasingly we seem to also find small pieces of fiber and other micro beads and things like that in our plankton samples. So that's something we are trying to investigate as well. And then last but not least, we have been working with Marine Life since pretty much the beginning of the survey, who send 2 observers every year who help us to map and quantify the top predators in the area in the autumn. So this is the marine mammals, the birds, and bluefin tuna as well. Over the years, the survey has expanded. In the very first iteration, we only surveyed the UK waters around the Cornish Peninsula. Then we got a bit more money and managed to expand that into the French waters as well, which was important for some of the species we were interested in. And in the last few years, we've had extra money from the Welsh government, who have asked us to extend the survey further northwards into the Cardigan Bay area. That was not the case last year, by the way. So we just focus on the French and English waters and Welsh waters around Cornwall. Okay, so first some results from 2024, starting with the oceanography. So this is all in situ data that we collected during the survey, right, in October. So what you see on the left is the fairly typical sea surface temperature map of the Peltic survey. So we have sort of warmish waters in the eastern part of the survey, both in the Western Channel, but also in the Bristol Channel. There's a typical cold water pool that we often see sitting out here off the North Brittany coast, and then further west in the deeper water, we get we get some sort of warmer temperatures as well. The central map is the sea surface salinity, which is perhaps less exciting. It's more homogeneous, as you would expect. Typically, we find fairly high salinity throughout the survey area, with notable exceptions in the Bristol Channel. Well, of course, the River Severn is responsible for an influx of quite a lot of fresh water. And similarly, riverine outflow is also responsible for Lyme Bay reduced salinity there, some slightly brackish conditions. The strongest salinities are always in the sort of deeper waters here, it seems. Now, this figure on the right is an interesting one, in my opinion, at least, and it's delta T, so it's effectively a map where we subtract the surface and bottom temperatures from each of our CTD stations. And what you get a map which shows areas that are where the waters are very well mixed vertically, compared to areas where there's what we call stratification, where the surface and bottom temperature are more different. Now, the well-mixed waters are indicated in purple on the right here, so largely most of the Western Channel and the Bristol Channel are very well mixed. And they get the stratified waters further west. Now, this white line is what we call a front, and it's an important boundary between these two areas. And we believe it's important for driving some of the ecology that we encounter during the survey as well. So here's a map where we've sort of summarized, if you like, the position of the frontal system in the survey area from between 2012 and 2021. And what you can see, there's what we call the Celtic sea front over here, which is pretty consistent from year to year, with quite a bit of pretty consistent basically. And contrast that to the ocean front at the mouth of the English Channel, which changes in position quite a lot from year to year. Now, I do point out, of course, that the time of the survey is October, which means it's a period of change. And if, for example, the autumn storms come earlier one year, they will mix the water early than so the scenario changes quite a bit from year to year, depending on when the storms are. We may sample at the same time, but the ecosystem may be at a different stage, if you like. So the oceanographic products I wanted to show are these ones. So on the left is the average temperature collected during Peltic, so October from 2013 to 2023. Again, as I showed before, you got the warmer waters in the east, cooler water in this North Brittany area, and then slightly warmer waters further west. Now, why is that interesting? Because if we want to know how 1 year compares to the time series, we can create an anomaly map. So that's the map on the right. And what we've done effectively is we've subtracted the 2024 temperature map from the long-term average. And any areas indicated in blue are slightly colder than the long-term average, and any areas in red are slightly warmer. And so if you squint your eyes, you could say, well, there you go. In 2024, the Western Channel was slightly cooler than the long-term average compared to the Bristol Channel in the north of the peninsula, which was slightly warmer. And that is true, certainly for that October period, although I would add that the temperature difference was only marginal, 0.5 up to 0.5 of a degree, which is not much compared to some of the years that we've surveyed, like 2023, for example, was very, very warm. Right, moving on to the ichthyoplankton. So this is fish plankton, literally. It's As I mentioned, primarily of interest because of sardine, which finds its peak spawning period at this time of year. So what we do is we count the eggs, we stage the eggs because that tells us what stage of development it is and how long ago it would have spawned. And we measure the length of the larvae again, so that we can sort of back calculate when they roughly would have spawned and where it would have been. So this is not a uncommon sort of picture. The results here for 2024, loads of spawning of sardine around the Mounds Bay area and the Isles of Scilly. Note some spawning also a little bit further north, interestingly, but very little in the eastern part of the survey grid, and very little on the French side as well, relative to here. Larvae, similar picture, but slightly smooth, if you like, which of course is understandable because these are slightly older, so they've had a chance to drift a little bit further. So, but yeah, the peak spawning is very much around the Cornish, the sort of western part of the Cornish Peninsula and the Isles of Scilly. Okay, top predators. As mentioned, since 2013, we've been working closely together with Marine Life, who every year, most years at least, send 2 observers and providing us fantastically useful information on the top predators. The fact that they found a few Balearic shearwaters in the early years drew the attention from Natural England and JNCC, and as a result, they've sent additional observers using slightly different recording techniques since. Balearic shearwater is the only critically endangered seabird in Europe, so it's drawn a lot of attention. So any sort of data on that is very important. But more generally, the collaboration with Marine Life has meant that we've got hugely important information on spatial, temporal trends on all these top predators. And an example of one of the products that we produce every year is this map on the bottom right, which is the common dolphin sightings. The little bubbles that are size dependent, correlated to the number of individual scenes, so bigger pods, smaller pods, etc. And we have this for all those years. This table on the left shows another interesting result. So these are the standardized bird observations by year for these years. So 2022 is missing for a reason I won't go into, but for the last 4 years, we see little trends here. So gannet's not surprisingly the most abundant bird species we encounter. But what may be surprising is that in 2020, when we had nearly 200, that value has dropped to just over 100 per unit effort in 2024. So a big decline there, which is highly concerning. Another perhaps more positive note is the interest of Cory shearwater, who we not encountered at any time for about a decade until 2023, and in 2024 was the second most abundant bird species we encountered in the area. So, you know, some good news stories, some bad news stories, but in my opinion, a fantastically useful data set there that we've only just touched the surface of using. Now that's talking about the trends and the sort of spatial information, but it's also thrown up some interesting anecdotal information. This is a picture taken by our lead observer, Pete Howlett, a few years ago. It's a gannet, for those who are not familiar with, but they should normally have a sort of pale iris, but as you can see here, it's black. And that later turned out to be associated with bird flu. So, but this was actually a surviving bird of bird flu, apparently. So they're, you know, really interesting sort of anecdotal, opportunistic bits of information. Okay, pelagic fish, my sort of main area of interest, certainly, and the one area that I'm going to talk a little bit more in more detail about. So for those who are perhaps not familiar, I've mentioned echo sounds a few times now. It's based on the concept that a pulse of sound is emitted from the ship and comes back through the water column and contains all sort of information about the organisms in the water column. And it's visually displayed like in the form of an echogram, which is what you see here. So it's a vertical slice through the water column, if you like. Seabed here, surface there. And what you see here is a large spawning aggregation of sardine in this particular case. You can imagine, though, it's sometimes hard to identify what these species are that we're looking at. So we do still deploy a trawl, take a small sample, which then gets sorted, split into species, and then we take a bunch of biological information parameters in our fish room, as you can see happening here. So really interesting information. These are otoliths, by the way, slightly different shapes for some of the key species that we look at. The otoliths are the ear bones, which tell us more about the age of the fish that we're looking at. But combining the biological data from the trawl with the echograms allows us to map the density distribution of the small pelagic fish community by species, and that can be converted, if you calibrate your echosounder, into abundance, basically, and that goes into the stock assessments. So let's look into a little bit more detail to some of the core species. These are sprat in the background, small silvery forage fish you may have seen before. That's what they look like when you bring a trawl back up. The top right are two examples of what they look like in one of those echograms. So on the left is an echogram of juvenile fish, as we see large, primarily in the Bristol Channel area. So they're sort of mid-water, smaller schools. Picture on the right is a rather large school, and I have to admit it's one of our best examples, certainly in 2024, of a very high aggregation of sprat in Lyme Bay. This one was completely surrounded by tuna who were frantically feeding on this very school. So what do we know about sprat? Well, there's a small-scale human consumption fishery targeting this, primarily in Lyme Bay, operating from Torquay. When we started this work, it was one of those data-limited stocks. There was no information really available on it. But through the work with Peltic, we managed to collect enough data on a number of different elements, which helped eventually to create a much better stock assessment for this stock, basically. That's what this paper is about. We did some interesting genetics as well, but I won't go into too much detail there. From an ecological perspective, this is a map of the density distribution of sprat, very much like I showed you before. So this is based on the acoustics. This is very typical for sprat. This is the 2024 results, but what we see is we see high densities in the Lyme Bay area. We see high densities also in the Bristol Channel area, and we sometimes get some further out into the western part of the channel along the southern Cornish coast. We rarely see them much beyond that. Occasionally we get some small numbers of sprat near the Isles of Scilly, but really not very much at all, and neither further offshore or along the French coast. So very localized species, very much so. The biomass fluctuates hugely. So this is the biomass for the Western Channel component only, this feeds into the stock assessment directly. And you can see strong fluctuations from 90,000 tons down to about 16,000 tons, then stabilizing more or less at 40,000 tons. And then we get this strange spike in 2021 where the biomass nearly quadrupled from the year before, largely driven by O group fish, completely gone the next year. No idea why, but interesting nonetheless. One of the more also interesting and arguably concerning observations, certainly from a fisheries perspective, is that the size of the sprat that we encounter in the Western Channel has decreased. And this is not that this is primarily driven by the absence in recent years of the large fish. So these are the numbers by age for 2024, and as you can see, loads of O group fish. So these are fish that are born that year, with much smaller numbers of H1s and even fewer H2s. But we would normally expect fish up to 5 years old, and we never encountered any of them. So whilst it's good news that there is strong recruitment coming through, so lots of babies coming into the population, the absence of the very large fish is certainly not good for the commercial fisheries, who are specifically focusing on those for the human consumption purposes. Right, next species, sardine. As you are probably aware, the pilchard, as it is also known, has been targeted by fisheries for many centuries around Cornwall and Devon. Most recently, the Cornish Sardine Management Association have rebranded it as Cornish Sardine, and have, with their pretty sustainable fishery, looked at the fish in the area using their ring nets. What's perhaps interesting for some of the paper that was produced by Southwood and colleagues in 1988, they looked at anecdotal information on sardine in the area, landings and things like that, and they found that there are warmer periods when sardine are more prevalent in the catch, and during colder periods, sardine disappears and herring takes over. So we know that historically, certainly the Cornish Peninsula and Devon have been at the northern boundary of the species. In 2017, it became a data limited stock because it was split from the Bay of Biscay population with which it was previously linked, which was a problem, of course, because data limited is of concern basically. So with Peltic, we had just started to work in the area, starting to get some biomass estimates for sardine. Ecologically, this is very much this is the 2024 results for sardine. It's widely distributed. Highest densities again in that sort of Mounts Bay area, Isles of Scilly area, southwest of the Cornish Peninsula, but it's found much further into the channel and also even in the Bristol Channel, interestingly. What's also interesting is that we have evidence that the full life cycle of sardine is found in this area. So there are eggs, there are spawning, and there are juveniles, nursery areas in between. So it seems to be no doubt migration, but there's definitely a resident population of some sort in there. And the biomass is high. So these are the biomass trends by year on the x-axis for two areas. So as I mentioned before, we originally started in a much smaller area, which is the area indicated in red here, which is that red line, and the more relevant area in blue, which is the one that feeds into the sardine stock assessment. And that shows that, yes, there is fluctuation, as you'd expect for these small, short-lived species, but the biomass up to nearly 500,000 tons is rather significant. So that's really good news. And that led to, as I already sort of briefly hinted at, to an assessment. So we needed an assessment following the split from the Bay of Biscay, which we successfully contributed to with this survey. And we're now further improving that because the time series at the time was rather short. So this is work in progress, but we did manage to contribute to that. I wanted to show you this map, or series of maps rather, which is acoustic density from Peltic, but combined with that of surveys at the same time of year in the wider Northeast Atlantic. So basically from Gibraltar to Ireland, this is the sardine distribution in the autumn. And there's a couple of things I want you to look at. So first of all, if you squint your eye and you look at it's effectively heat map. So the areas that are lighter are at the highest densities. And you see that Cornwall and the waters in the Western Channel are as important to sardine as some of the more traditionally well-known sardine areas of Portugal or further south. So that's really interesting. The other thing I wanted you to focus on is if you look at north of the Cornish Peninsula in the early years, and this starts at 2017, of course we have data from before that, but we found very few sardine north of the Cornish Peninsula, a little bit, but not huge amounts. Whereas in the last few years, 2024 and 2023, that's much more prevalent. So there seems to be a northward expansion. And that's also reflected by the egg distribution. So these are the eggs from Peltic collected between 2012 and 2021. The top row is the years in which we only covered the UK waters. Again, I'll keep reiterating it. Whereas the bottom row is when we had the full coverage, if you like. And in the early years, we had 0 eggs north of the Cornish Peninsula until in 2015. From 2015 onwards, we started to get eggs there. So really interesting there. Spawning activity seems to expand further north as well. Acknowledging that, of course, this is a snapshot and we don't quite know what happens before or after, but still interesting. And then the final point I wanted to make about sardine is some genetic work. It's a complicated species genetically because it's quite migratory. So that means any genetic signals tend to be really faint. But we did publish a paper which confirmed that there was that basically there was an independence of sardine from the North Sea and the Bay of Biscay, which is not completely unexpected, but there's a geographic relation there. So the further apart, the more different they are. That's not uncommon for migratory species like this, but a follow-up paper which looked into a little bit more detail suggests that at least genetically, the Eastern Channel and North Sea are more similar compared to the waters to the Western Channel. So That's only the genetics, and there are other elements to consider when you want to understand population dynamics. But yeah, it's interesting nonetheless. Good. Anchovy. The last species I'll delve into a bit more detail. In fact, quite a bit of detail on this one because it's possibly one of my favorite species. It's quite a dynamic species. This is a map from 1883. And in red, it indicates where anchovy are. And surprising to me at the time when I found this map, it's been in and around the British Isles and southern North Sea before, right? So certainly that wasn't the case when I started looking into anchovy. And as far as I could tell, from the 1930s onwards, there were no anchovy, or very, very few anchovy, north of the Bay of Biscay, basically. That was really very much the northern distribution from the 1930s. Now that changed. In the mid-1990s, there were increasing scientific papers showing anchovy showing up in the North Sea, some of the bottom trawl surveys. Anchovy was occasionally caught by some of the fisheries operating on the Cornish south coast, completely unexpectedly. And so this period in the mid-90s was quite peculiar, quite different from what had happened before. And of course, the big question was, where do these anchovy come from? Are they the Bay of Biscay fish that have moved further northward due to climate change or whatever? Or was this perhaps local population expanding? Because as we've seen on this map on the right, they were there previously. So that was the focus of a paper that was published in 2012. And it studied, it tested very much these hypotheses. Is it local populations that have somehow clung on in very low numbers in the southern North Sea, perhaps? Or is it Bay of Biscay fish, either juveniles, eggs, or perhaps adults that have migrated there? And they adopted a multidisciplinary approach, using genetics, looking at bottom trawl surveys, but also modeled larval drift. And they found in that paper that no, there's strong evidence that Bay of Biscay anchovy cannot have made it into the channel. So pointing towards the fact that these are local remnant populations still alive. So we had a scenario here where in the north, we have this spawning activity in the southern North Sea, and those fish would migrate into the channel to overwinter in the western channel. So those would be the same population, and there's a separate population in Bay of Biscay. So these green blobs are spawning areas. In the autumn, these northern fish migrate into the channel area, western channel area, and the drift showed that eggs spawned in early spring in the southern Bay of Biscay, where at this time spawning took place. They could drift a little bit, but later in the year, the prevailing currents would be southward, so they could never make it in time to the eastern channel, right? I want you to remember that, because we were quite content with what we're doing in the Peltic survey. We knew now that there were two stocks. There was one in the Bay of Biscay, which was already managed very well with loads of different surveys. And we now started to build a time series with Peltic on the northern anchovy. So these are the fish spawning in the southern North Sea and overwintering in the western channel. So perfect timing for us to be in the channel. And this is what we saw. So again, 2013 to 2021, in green anchovy distribution, and the bubble size is relative to its abundance. And if you squint your eye, and acknowledging that, of course, in the early years, we only sampled the UK waters, but if you bear that in mind, we see an expansion of anchovy. Anchovy is found further west originally, then also further in the Bristol Channel, and we really see an increase there. And in fact, I remember the anchovy that we caught in the trawls used to always be mixed in with other schools like sprat and sardine. But in more recent years, we saw dedicated anchovy schools everywhere. This is a 25-meter example, just one of the many examples we saw in 2023. And I also would like to point out that we, of course, sampled the ichthyoplankton as well, and there were no eggs of anchovy in the channel during this period at all. Biomass has also increased quite a lot, so the bubble size suggested. And in 2023, we had a 250,000 tons. So that's a 5-fold increase compared to 2021. Completely unexpected. And unfortunately, in 2024, we barely found any anchovy. So again, one of those short-lived species traits that to peak and then crash again. But anyway, biomass has been steadily increasing over the years. Then in 2019, and even more strongly in 2020, we found something really strange. We saw these schools at the surface on the French side of the channel, so the south side of the English Channel, which were strange. We'd never seen those before. And when we trawled on them, we found them to consist of very small anchovy. So just for reference, a picture of them here. So these are post-larval anchovy. They've only just started to get adult shape and color. The bottom one here is not even got its silvery coat, if you like. And what you see at the bottom figure are the length frequency histograms of normal anchovy that we would have encountered, which are those northern fish, right? So from, let's say, 8 centimeters upwards. These fish in these small schools near the surface were much, much smaller, completely different compared to anything we've encountered. And this happened for a few years going. And when we looked at it, they seem to sort of be distributed all along the French coast. Some years not as pronounced as this, but this was a good example. And it really looked like these very juvenile fish were migrating from the Bay of Biscay. But how does that work? Because we know that wasn't possible, according to the Pierre Petit paper. But yet they're smaller than the anchovy we normally encounter. We know there's no local spawning. So what's happening, right? So, sorry, this is the Pierre Petit. So yeah, no, they can't make it. That's what the drift modeling suggested when we looked into that. So what's happened? So basically, we published a paper last year in which we summarize this very phenomenon. And we again used multiple approaches. So there's the genetics. We had genetic samples of these fish, and we can quite clearly distinguish the northern anchovy, if you like, from those in the Bay of Biscay. And we looked at any changes, particularly in the Bay of Biscay, that may have been, the cause behind these observations. And what we saw is that when we looked at the spawning activity, that spawning in the Bay of Biscay has been expanding both in space and time. So there's more spawning later and later in the year, and also further north, which meant that this southward current that prevented previous larvae from making it to the channel was no longer impactful because the fish were already further north anyway, so they could make it around the corner. So in summary, we had a scenario in the mid-90s where after absence of anchovy in the channel northwards, we suddenly started to see anchovy. We're pleased this green spawning area is where they spawned, with the nearest spawning area much, much further south, no spawning in between. Two separate populations, separate management units, and really interesting. Then, because of a change in spawning area, which we believe is due to the fact that the anchovy population in the Bay of Biscay has done really, really well. So there's so much density there that they've sort of expanded in space and time. And that's the cause for these new observations. Probably linked to milder and warmer conditions, which enable them to expand in space and time. Now, this is interesting, but of course it becomes a bit of a nightmare from a stock perspective, because where they had previously been separated quite clearly, that is now no longer the case. But it's also interesting from an ecological perspective how different mechanisms operate here. Okay, I promise I'm getting towards the latter stage of the presentation. Just wanted to mention some of the other fish that we regularly encounter and do collect biomass data from, certainly the top few. So herring, I didn't present in more detail because we don't get that many, but there's always juveniles in particularly the Bristol Channel. The area is really important nursery area for both horse mackerel and mackerel. And in the bottom right, you see a map of boarfish, one of those quite attractive looking little bentopelagic species that occasionally find themselves in big waves coming onto the shelf. And it seems that right around the Isles of Scilly is where the edge of the distribution is. So proper deep water fish, really interesting. And then unfortunately no biomass estimation, but we do also get saury pike and garfish in our survey. Only in the trawl, really. They're quite hard to monitor, but we've been able to collect quite useful biological information on these really important prey fish for a lot of organisms in the southwest. A dedicated slide to bonito, a species that we first encountered in 2018. Yes, they had been present in the area, and they still are present in the Western Channel during the summer. But this was the first evidence that we are aware of, at least, that they try to overwinter. And we thought this was interesting, climate change. But beyond 2018, it only happens again in 2020 and 2021, and we haven't seen them since. So who knows? I mean, we may have just missed them because we don't trawl that heavily. But yeah, to be continued. And also in 2018, interestingly, we were looking at the agrogram ready to start our transects at first light, but we were a bit early, so we were sort of hanging about to the southwest of the Isles of Scilly in about 120 meters of water. So there's quite unusual schools mid-water, which as the sun started to rise, started to drop down in vertically and started to become more fuzzy. We've never seen that before. We managed to get a small dip with a troll in it, and didn't catch much at all, but had a few pearl sides or lanternfish in the mesh. And for those that are not aware, this is one of the most abundant vertebrates on the planet. This group of what we call mesopelagic species. They're normally found in the deeper waters off the shelf edge, but for some reason in 2018 they were on the shelf and close to the Isles of Scilly. So never seen it since. We had the occasional individual, but yeah, really strange. And then finally, from mesopelagics to large pelagics, bluefin tuna. I just wanted to mention briefly that the data collected by the observers has been really instrumental in providing the evidence that this species returned. First observed in 2014 in the Celtic Deep, and then in increasing numbers ever since, certainly in those early few years, as nicely written up by Tom Horton, using data from a lot of other organizations operating in the area, all of which corroborated this pattern of the return of bluefin tuna. And whilst the drivers of this are not 100% confirmed, it's likely to have to do with the population in the Mediterranean doing quite well. But of course, they wouldn't be here if there wasn't any prey. So the high diversity and biomass is probably something to do with it. Okay, so this is I just wanted to summarize the sort of main impact of 12 years of Peltic from a pelagic fish perspective. So we managed to contribute to 2 stock assessments, one much improved stock assessment and one brand new assessment for sardine. As I said, that's one that we're continuing to work on to improve it further. Should there be a need for an anchovy assessment, we have the data to underpin that, so that's good to know. I haven't really mentioned the fisheries management plan, but the Peltic survey has contributed to the evidence documentation on both the spread FMP and the Celtic Sea and Western Channel FMP. From an ecological perspective, I've been finding it mind-boggling that we get these crazy spikes in biomass from year to year. So different years, I didn't mention the herring one. It was only relatively high, but it was quite noticeable. Yeah, from one year to the next, quadruple biomass, and then drop to normal again the year after, to the more average. For spread, the most notable change really has been the change in this age composition. So we're lacking those sort of older fish, but the biomass looks healthy. For sardine, interesting evidence now that the full life cycle is completed in the Western Channel and the wider area. They seem to be increasing their distribution, potentially moving further northwards. It's just a visual assessment, to be honest, but who knows? And yeah, also spawning north of the Cornish Peninsula seems to be increasing. And then I won't repeat the anchovy story because I've probably done that ad nauseum, but yeah, quite a lot of crazy events going on with that species, proper different mechanisms causing it to increase in our waters. And I pointed out some of the ad hoc events that we've been able to capture with the bonito and the pearl size and the return of the bluefin tuna. So I'll leave it there. If you're interested in reading more about some of these details, a lot of the sort of stock-related work is published on the ICES website in these 2 working groups, but many others as well. If you're interested in the more science components, we've actually created a Google Scholar page for the survey, in which you can find all the papers that I've used for this presentation, but also many more which I have not been able to cram in, including that on diet and ecosystem modeling exercise we've done. One particular paper I wanted to point out, written up by a colleague who looked into the smaller components of the ecosystem, all the various plankton sizes, and compared two very contrasting years from the survey, identifying very interesting patterns. So I'd encourage you to look it up if you're interested. And then data. I mean, perhaps if you find this sort of information interesting, we publish the reports on an annual basis. They're all publicly available on the BODC website a couple of months after the survey completes. And then the various data sets are available at these sources, all publicly available. And if it's not, then and you're interested in something, just get in touch, really. So thanks very much for your attention, and I'll now take any questions. That's eerily quiet. Shall we? Does anyone have any questions?
I think we're it's kind of stunned silence, really.
I've put everyone to sleep, maybe, but
yeah. No. It's far from going to sleep. It is exactly why I kind of did, why the conference was delayed 15 minutes over lunch.
Okay.
You have a few hands up, Jeroen.
Yeah, great.
Yeah.
Let's look at the order. I think I should be able to figure that out. So, Kaylee, you're first. We've emailed before.
Yeah, thanks for a great presentation. It's really nice to see how you can monitor a whole ecosystem. I was just wondering, you mentioned eDNA at the start, and I was wondering what you're doing with that at the minute, or are you kind of just testing methods?
So I can tell you, and actually I was half expecting a question on this. So I've got a slide. Here's one.
Oh, wow.
Yeah. So it's early stages, but we had a PhD student. So I explained how the acoustic methodology is the key one to use for small pelagic fish. It monitors the water column really well, but its biggest downside is probably the identification of species, right? So we deploy trawls largely for that reason and for biological samples. Now, at least the species identification, we figured, could potentially be provided by something like eDNA potentially. So that's the route we're taking. So we use eDNA semi-quantitatively, acknowledging the fact that, for those who are not familiar, by the way, eDNA is the concept that a bit of DNA is left by an organism in the water column. You take a sample of the water, you filter it, you replicate the DNA to a slightly larger volume, and then you can cross-reference that to existing libraries of species that you know are there to identify which species are there. So it's good from a diversity perspective. There are still issues about its quantitative merits. Does more fish mean more DNA? And we know it's not that simple. So this is where we figured we use the DNA still the acoustics still quantitatively, but the eDNA semi-quantitatively. So that's sort of whistle stop of that. What you see here are an example of the 2018 survey where in the top we've partitioned the acoustics purely by the trawl. So you see sort of core densities of spread in Lyme Bay, in the Bristol Channel, pretty much like I showed you before. Interestingly, most herring in the Bristol Channel. The bottom is done the same, but use the eDNA instead of the trawl, right? So similarities for sure. So you get that big density in the Bristol Channel, but funnily enough, Lyme Bay spreads. Pretty much no eDNA found there. It's all found further to the west. And interestingly, in contrast to that, herring in Lyme Bay, hugely dense, whereas we didn't catch any in the trawl clearly. So yeah, early days, but that's how we apply it. So I stop sharing there and let you actually get in because you may have a question, more further question.
Yeah, that's great. Thank you.
Is that all right? Yeah. Yeah. Cool. Right. Who's next? Alex.
Hi, Jeroen. Good to see you.
Hi.
How are you doing? Well.
And you? Yeah, and you.
You can guess what I'm going to ask. How do we get hold of the bird data? Because I still chair the SWME bird group, and we have quite a lot of observational data on stuff like Corrie shearwater, but we don't have a lot of at-sea data. So it'd be really valuable if we could access it.
Yeah, that's great. I mean, I'm very keen to make them available. We're sort of trying to manage it to a degree in the sense that we'd like to look at some of the ecological questions as well. But yeah, do you want to send me an email afterwards?
Yeah.
Copy Tom in as well.
Okay.
And Tom Barreton, because we sort of co-own the data, and we can take it from there. But you may want to speak to JNCC colleagues as well, because they've done effectively what you did in the early years, which is using the ESAS methodology. So that's still alive as well. I don't know about sharing of those data. That's for them to decide. But yeah.
Okay. Sounds like we can resolve it. That'd be really helpful. I'll drop you a line. Thanks, Ron.
Nice one. Cheers, Alex. Bryce.
Yeah, yeah, thank you. Fantastic presentation. Really, really interesting. I'm interested in the tuna. I might not be the only one. I guess there's all sorts of questions about how their distribution and abundance being driven by these changes in prey. But I guess I was wondering how you guys are actually trying to count them. Having spent a bit of time at sea looking at tuna schools, it's somewhat overwhelming when you see them.
Yes, quite. And I'm probably not the best person to answer this, in the sense that the marine life experts do that for us. And when they do, we're all very much aware that it's there are certain caveats to the methodology. So they're very good at doing it for dolphins and things like that. So they use similar methodology, but of course dolphins need to breathe at the surface, and tuna don't. So having been on vessels that troll for tuna, I've seen tuna biting when there was none to be seen on the surface, and I've seen tuna on the surface when none bit the troll. So yeah, it's very much we keep doing it because we're out there and we do observe. We're very, very aware that there's no. I mean, interestingly, Tom Horton, who wrote that paper on tuna using these data and others, he found quite interesting relationship between the number of observations and the biomass trends predicted, calculated for the Mediterranean stock. But that was a very short time series. It's very likely to fall apart if you extend that to longer periods. But yeah, it should probably not be used quantitatively, but it's better than some of the information that we have, right? So
yeah. I mean, it's better than nothing. Are you able to tell me who to talk to about it?
About what? About the methodology? Yeah.
Yeah, or who, somebody who's collecting the data on them.
So the marine life colleagues own those data. We share them with us. So we have.
Okay.
The methodology is probably best described in that Tom Horton paper, by the way. So there's different categories.
Okay, all right. I'll have another look at that.
Yeah. It describes how we classify the certainty of the observation. So probable, those sort of things. And was it a single splash or was it a proper, feeding frenzy sort of thing? So those are probably the best first steps, really.
All right. No, brilliant. Thank you. Yeah, really interesting. Cheers.
I'm glad to hear it. Thank you. Cheers. Hi, Duncan. You're next, I think.
Sorry, just miking as well. Hi, brilliant presentation. I really enjoyed the last year's one as well. I think it's really good information. I know you're talking about what you've collected on the Peltic cruises, and it's really interesting to see the increase in sardines and the possible northward shift. I was just wondering how much information we have about sardines in the past, because obviously there was a big sardine fishery.
Absolutely.
And the fishery disappeared, and there were economic drivers and other things like that. But so now we're seeing this big surge. I was wondering if you had any understanding of how that relates to what has been in the past.
Yeah, really good point, Duncan. I mean, when I talk about change, my field of view is 12 years, right? So we may have some information from before that, but not collected at the same methodology. So drawing any conclusions out of that is dangerous. And some of the anchovy example is a good one in the sense that we thought they had not been here, and of course they were. It's just that periods in the past were different. So we don't. I have some ideas of what may have happened. We have some data actually. I found some historical data from the beginning of the 20th century. Sorry. So not going back quite as long as the fishery, because that was obviously many centuries old. But I'm planning to look at some of the data that we have in the lab stored in these old historical containers. It includes age data, but so we should be able to deduce some features out of the population from that. But quantitative, it's going to be really hard. I mean, the best effort that's been done on that is by Southwater and colleagues in that 1988 paper where they looked at landings, including anecdotal stuff. So I'm not going to try and sort of improve on that really, because they did a great job, and I don't think better data are available. So what I will focus on is some of these scientific data from both commercial catch, but also from our institute, which were collected going back 100 years or so. And I'll that's very much a plan. When I'm going to do it, I don't know, but that's at some point that's on the list for sure.
Excellent, thank you.
Cheers, Duncan. Sue, you're next, I think.
Okay.
Oh, no,
hello, sorry.
Bob, sorry.
Sorry, Bob. Go for Bob.
Yeah.
Okay. That's very, very good. Thank you. In thinking about the fish chapter, and I hope Doug Hurton will kind of come in with this, and some of the things that we picked up in the fish chapter so far. I was interested to see your kind of additional comments on garfish and Saury pike, because we'd had a debate in the fish community of practice, swimming fish community of practice, about which species was involved or whether it was both. So that was it's interesting to hear you confirmed that it was both.
Yeah.
There was a similar conversation about bonito, which we've got in this fish chapter already, with some records that were taken in a benthic trawl in the winter in the North Bristol Channel, which it seemed kind of rather odd. One of the things we're also commenting on, which I think is a standard thing now, is it's very clear that mackerel are arriving much later in the season, if at all. And I do remember going back sort of 10 years or so ago, you showed this picture of a massive mackerel shoal in the whole channel, which went from virtually one end of your transect to another. So I wonder if you could actually sort of say something about that. And then, I mean, so in a certain sense, I wonder if you'll scan the fish chapter for us to kind of provide a kind of editorial. I mean, Doug's here as well, so I'm sure Doug will have picked up on a few of these things as well.
Right. Well, thank you, Bob, for those questions. Bonito first. I mean, I'm more than aware that certainly our data there's an element of chance involved in it, right? So we don't fish as much as any fish survey does, because we don't need to. We have the acoustics to quantify it. So our fish sampling is quite sort of targeted and sporadic. And I suppose if something is really rare, like a bonito, then there's an element of chance of it being caught by the trawl. Of course, that increases with the number of fish. So I'm not surprised there are examples of bonito in the winter. And that's for that reason, right? So there's really interesting still to hear about it. In terms of the mackerel, you remembered exceptionally well. In 2013, we had a school that was, if I recall, 30 miles long, 20 miles wide, and about 40 meters tall at its peak. And that's reminiscent of some of the mackerel schools that Lockwood wrote about in his mackerel book in the Southwest, but we've not seen it since. And in fact, if anything, we've seen a sort of decline in the number of schools. Certainly, it's acoustically slightly more challenging to deal with. So I can't be more clear than that. But yes, I have all sort of other theories about mackerel, and its importance of the southwest as a nursery area as well. By the way, but I think this session is probably not long enough for that. So, but yeah, I know Doug probably has comments about that. I do wonder whether we should do the order properly. Would you mind holding on, Doug? And
yeah, sure.
yeah, and then because I'm sure you're responding to that. Sue has been having to wait. So yeah, Bob, and the editorial I can do. But the one final thing I was going to say is that of course my views of the area is very much a snap of that of Peltic, which is a snapshot, right? So I have ideas, but of course there's people locally who have a lot of knowledge of what goes on through the rest of the year, and I'm sort of more than aware of that. So, but yeah, happy to provide some feedback if that's of any use. So, Sue, finally.
It's all right. I'm happy to wait if you want to go straight on to Doug, although I'm sure Bex won't mind either. But does Doug need to respond to that? Is it relevant?
Well, it's okay. I'm coming after.
Okay, so I'm assuming that marine life records seals as top predators, and we've seen a shift. So I'm from the Seal Research Trust, and we've seen a shift in our surveys from seals moving west from the north coast, from the big sites on the north coast of Cornwall, west round Land's End, and onto multiple smaller sites on the south coast. Now, clearly that's likely to be fish prey driven, and if so, do you have any theories about that?
Wow, very interesting. And I'm probably going to have to say I don't. I mean, the marine life do record seals. You're quite right, but the numbers are exceptionally low during the survey, just because our interaction with the shore, where they're obviously more prevalent, is very briefly. You saw those transects, right? So yeah, we don't have any. I wouldn't say we have any useful information in our observer data there. In terms of the migration around the corner there, that's interesting because a lot of stuff we're monitoring seems to, expand northwards, right? So, I mean, seals eat not just small pelagic fish, right? Which is of course the species that we're primarily targeting. So I can imagine that and there may be shifts in demersal fish, some of which will be very suitable prey, right? For, I mean, I'm not an expert in pinnipedes, but yeah. So I'm just rambling here. I'm not sure, basically.
No, and we don't know very much about benthic fish species either. But given that kittiwakes are declining in Cornwall, if that means that sand eels are declining, which I don't know whether it does or not. It's just I have personally had a theory that it might be related to the anchovy moving north, and I'm assuming that seals are not moving north, are able to move south because of their body's ability to adapt thermally to a wide thermal range. But yeah, it's just fascinating. Things are definitely changing, and obviously fish is changing too, so it's not really surprising. Thank you.
No, and thanks for your question and comments. You're quite right. It is very interesting, and there's a lot of change happening there. So, yeah, very interesting. Thank you, Rebecca.
Hi, thanks. That's really fascinating. I'm from the Cornwall Wildlife Trust, so I oversee the kind of Sequest Southwest survey cetaceans and things. So I was just wondering, do you have an overall sense of biomass of all these bait fish if you lump them all together? How that's changed, because you're saying year on year out, like you have different proportions of the species, but I was just wondering, have you seen a change in, like I said, if you lumped it all together as one biomass of tasty fish, would that give you any patterns?
Yeah, well, the overall pattern in the last 12 years would have to be an increasing one. I would have, I mean, I could easily, I suppose, do this exercise and sum them all up. The species I showed you, spread, sardine, and anchovy, are the dominant ones there. Really, I mean, horse mackerel and mackerel. Yeah, like I said, mackerel. It's a bit difficult to say, but yeah, the biomass will not be anywhere near that of those species. So just simply adding them up should generally show an overall increase, I would have thought. Yeah, I mean, what makes it difficult is that, of course, we've got these colder water species like herring and to some degree spread, which sort of compensate for. So so I suppose if there is a warming, you get prevalence of some of the warming species potentially compensating the demise, if you like, of the cooler water species. But overall, some of the biomass we've seen the last couple of years, 500,000 ton. I mean, I have to think about saying that nearly it's that high. It is very high. That's high for any ecosystem. So yes, I would say there's an increase.
We're sort of thinking we've seen a shift in sort of which species we're seeing since about 2015, 16 in terms of those large mobile predators. So it probably would help to explain that. Okay.
Yeah, it's fascinating. I mean, we'd really need someone to really apply some serious statistics at it, and it will be difficult because, of course, the time series is not particularly long, and and it's a snapshot, right? So what we see. I mean, certainly from a stock perspective and population perspective, I'm reasonably happy that captures the annual situation, right? Because that's what they're supposed to do. But some of the environmental components are more difficult to interpret in that context. So, but interesting. Yeah, thank you. Douglas.
Okay, several points. You talked about historical. In fact, I did look into a while back the anchovies, and found from the Marine Biological Association records from the 19th century of them turning up in Kent in the autumn, and one year, and then a couple of years later, somewhere further along the south coast. And they were thought at that time to be spawning in the Zuyder Zee, and the stocks disappeared when it was reclaimed.
Yeah.
And I wondered if the warming of Waddenzee might have produce the increase in the stocks we're getting now.
Yeah, very good point, Doug. And I'm sure you have more questions, but I'm going to butt in straight away. Quite right. The 1930s reference in that slide was very specific, actually. I didn't go into detail, but it was 1932 when the Zuyder Zee was sealed off, if you like. And you're quite right. There's very good evidence that the anchovy was incredibly high, the anchovy population there. Perfect sort of shallow conditions, warm up quickly. That's what they seem to like, certainly in these northern parts. So yeah, absolutely right. And there's a couple of German papers from the 1889, I think. And then, of course, this Piscatorial Atlas map, which is from 1883, which all confirm that in the late 19th century, they were probably everywhere. The whole Waddenzee, there's definitely evidence of spawning there, but surprisingly not as much as you'd expect, given some of the population increase that we've seen in the channel. So I'm still a little bit uncertain about it. I think they've expanded their spawning area. We have evidence of spawning in the Thames estuary now. We had some ichthyoplankton surveys there, which suggests it may be quite a significant spawning area, but that's been very ad hoc, so no trends there. Anyway, next question.
Yeah, and bonitos, finding several years back started to get commoner, especially around the Channel Islands.
Okay.
And also, I got the impression that years I got lots of records of bonitos were times when we got more records of anchovies.
Okay.
So that appeared to be a possible, there's very few number of bonito records, but even so,
interesting.
Lots of those. It was.
And it's often, sorry to interrupt you, Doug, but those perceptions, as you call it, are quite interesting, aren't they? Because that's how these ideas start, and we can then investigate it a bit further. I mean, 2018 was a peculiar year when we first saw the bonito in the survey. That's why 2018 I mentioned. But it was a funny year because, funny enough, also it was herring, one of our cold water species that did quite well. We had probably our biggest catches. So what exactly 2018? Where that fits into the picture, I don't know, but I'm certainly going to look at some of the links that you perceived between anchovy and bonito. Thank you. Yeah.
And the pearl sides, I did a short paper in the Isles of Scilly bird report on pearl sides because they turned up in quite large numbers, stranded on the shores of the Scilly Isles one year.
Oh, do you remember what year that was?
I can't say off now. I can send you a link to the paper.
Interesting. Yeah, please do.
There had been odd ones before and other ones before, but it was only hundreds, but still it was for strandings when there hadn't been many reports, none from there before that.
Yeah. Crazy. Yeah. Interesting.
Unfortunately, just as you were talking about sauries and garfish, I had a phone call came in and I wasn't able to follow you. The impression I was getting from talking to anglers and that is they see quite a lot of lots of garfish all around. I was down on the Mountbatten breakwater in Plymouth 2 years ago,
Right.
and they were just putting a line in the water and pulling out garfish and putting them by to freeze up for bait through the winter.
Interesting.
But very seldom hear of sauries. And I was wondering if garfish are more the inshore coastal population and sauries are more oceanic. Is that the case?
That would be my perception. But again, it's one of those species that we only pick up by chance, really, or group of species, I should say, because there's obviously multiple. But yeah, because of their position subsurface, we don't see them on the echo sounder. So the only times when we encounter them is when we happen to have gone through them, either deploying the trawl down or up, right? So
Yeah.
I would have to plot it up, but that's certainly it would be, yeah, that would match my perception as well. But it's an interesting question. We definitely get both on the survey, and so definitely offshore, more chance of scomberies of sorry pike. Yeah, good. I don't know whether we have enough data to do anything sensible there, but it's definitely.
I don't know if you've seen Rupert Kirkwood's photographs. He's got fantastic photographs of the tuna feeding.
Tuna, yeah.
And it always looks like garfish they're chasing now. I haven't seen it look like a saury, but it could be. Difficult to tell in photos, but.
It is. We have definitely seen saury being chased by tuna as well. Yeah. So including offshore, to be honest. So yeah. No, that doesn't exclude. But yeah, I'll have a good, interesting point.
Yeah, really fascinating, and I love the anchovy stories.
Well, I'm glad you did, because maybe it's a bit for the purists, but I just can't get over the fact that in a relatively short amount of time, we've had two completely different mechanisms driving a northward shift. So yeah, really interesting. Thank you. Right. Bob.
Okay. I'm looking at the screen. I just wonder whether we need just to kind of round up with anyone else who wants to ask any more questions. Otherwise, I'll say thank you with a couple of other plugs, if I may.
Go for it. Yeah, no, I think that's that was the last. The only hand still up is Douglas, but that's presumably remnant or whatever the legacy hand, I think they call it. Okay. So yeah, go for it, Bob.
Well, Jerome, thank you very much indeed for this mind-boggling presentation. It probably left all of us in a sort of slight state of fishy shock. Such kind of concept exists. It's really incredible. We'll follow up on a number of these connections. It's the connections that are really interesting. That the bird stuff in particular, Alex has already picked up on, which is really, really useful. Because we are kind of extending, the sort of bird side from the kind of terrestrial breeding productivity side to kind of more of these observations. And the observations of great shearwaters and Corey shearwaters over the last couple of years have been, I mean, stunning in terms of their kind of rarity and numbers. It's just ridiculous. But anyway, thank you very much. I'd just like to kind of make a point for anyone who doesn't know about the Southwest Marine Ecosystems. We've now established, Doug's led this for a number of years, but we've now got 50 or so people in a community of practice that are looking at the fish. If any of you would like to join that group, please contact Doug or Simon Thomas or myself, or Keith Scott, or any number of people you know, and we'll certainly add you to that email listing. We've got a draft fish chapter, which is fantastic, and that's relating to Jerome about. The reality is all we've got is snapshots from a whole variety of people, from the divers who snapshot, the bream and things coming into the waters, from more and more webcams that are left tracking what's going on, citizen science watching those, from the anglers who are picking these things up in a whole variety of different ways. Then there's eDNA coming along, and there's things like fish intel. So there's a series of snapshots, but collectively, we can actually begin to try and make sense of what's going on. And it's been great over the last kind of 4 months to sort of see how this kind of fish community of practice has come together to kind of pull this chapter together. It's been really very inspiring and very helpful. And your talk's been enormously helpful in that context. So yeah, hopefully we can make this a regular contribution. Now we've kind of cracked the kind of webinar side of this thing. And one of these days you might even come back down to Plymouth for the conference and things as well, which would be great.
I'd be delighted to. Yeah, if we could make it work, yeah, it'd be great. Yeah.
Thank you. So thank you very much indeed on behalf of all of us. I don't know whether Doug, you wanted to say something about this as well.
Yeah, I think that may be the legacy hand.
No, that's fine. Yeah, thanks so much. It was very, very interesting.
Yeah, thanks
indeed. Thank you. Thank your team for kind of helping out as well. And
Samantha, she's there. Thanks very much, Sam, for managing that. Yeah, cheers.
Sam, terrific job. Thank you.
Yeah, thank you.
I'll stop recording now as well if you're happy.
Yeah, we may have to. Yeah, we may have to do some editing, I think, but that's fine. Yeah, thank you very much, Sam. Much appreciated.
No, that's terrific. Yeah, fine.
Great. Well, thanks very much. Let's